Hello, today we are talking about something called transcription. Just as a review, we've been talking about DNA, about how it replicates, how it can make copies of itself and replicate. Today we are going to talk about how cells use DNA to make a product of RNA. How does DNA transfer itself into a product of RNA, and then from there into another product. So transcription is the very first piece of that, and we're going to talk a little bit more about that and how that works. So how is the code of DNA converted into some functional product? In the end, DNA, we start with DNA, it is converted into a protein. And how does this happen? This is a very much an overview. We've talked about DNA and the structure of it. We've talked about how DNA can replicate itself. We've also we are going to talk today about transcription. That is where DNA converts itself into an RNA transcript. From there, we're going to talk about in the next video how that piece of RNA, and it's called mRNA, how that leaves the nucleus and is, through the process of translation, turned into a protein. And then that protein carries out different functions in the cell. That's an overview of what we're going to be talking about. So why is transcription necessary? Remember, transcription is going to be the process of converting DNA into RNA. That RNA then leaves the nucleus and it goes out into the cytoplasm of the cell and does things. Why do we need to do that? What's the point? DNA itself is considered a long-term copy. It needs to be preserved. We do not want the DNA to degrade, to be altered, to be damaged in any way, believe it or not. Out here in the cytoplasm, if this is the nucleus right here where our DNA is housed, and this is the cell cytoplasm out here, we do not want the DNA out here being exposed to dangerous chemicals, different enzymes, things that could harm it and damage it, because that DNA needs to get passed on to the next generation. It's really important that it remain safe and preserved. So what happens? The DNA makes, sort of like here it's pictured like a CD-ROM or something, makes a little copy. And that copy then can, of RNA, that's the copy, it can go out into the cytoplasm. It can be seen as kind of portable or disposable. It's a temporary copy. And we look at that RNA as template as that temporary copy so that it can carry out what needs to happen to make proteins, to go into, do what it needs to do. It is easily broken down. It does not, it's very unstable. It's very short term, doesn't last long. So we can get rid of it. It can be processed through the cell and disposed of, and we don't have to worry about the DNA being exposed to something that would harm it or change it, damage it in some way. It's safe inside the nucleus, and we're just using this copy. So that is why transcription is necessary. That's why we want to turn the DNA into some RNA so that the RNA can then go out and carry on whatever needs to be carried on. And this is how that happens. That process itself is called transcription. And transcription is almost identical to replication. The only difference, we have an unzipping right here. It unzips in the middle. Here's our DNA strand, right? Long DNA strand. Unzips in the middle. We have base pairing, complementary base pairing that happens in here. There are a couple of differences between transcription and replication. One is that only one side of the DNA is used to make the RNA transcript. The other side is not used. Once the RNA transcript is made, it leaves and then the DNA zips back up. So that's really the only thing that happens. Another big difference, and you can see this in here, and we talked about it earlier when we talked about the DNA molecule. U is uracil, and uracil replaces the T for the thymine on as for the complementary base pairing. So where we have a T on the DNA, we will see a U on the RNA transcript. So that is the other difference, the two differences that we're going to see. So let's take a little time to review the differences between DNA and RNA. This is completely review for you because you guys have already learned this. Remember, here's DNA. It's a double helix. RNA is only a single strand. Because it's only a single strand like this, it doesn't have the complementary base pairing, it is able to fold up into a lot of different conformations. And so RNA, whereas DNA it has very specific ways that it folds up. RNA has a lot of variety in how it can fold up. We end up getting a lot of different types of RNA. We have mRNA, stands for messenger RNA. We have tRNA, rRNA, and even more, and they all function differently. Remember again, uracil replaces the thymine. Uracil is in RNA, thymine is in DNA. Another difference between them is that they have different sugar molecules on the backbone. So the sugar phosphate backbone for DNA has the sugar called deoxyribose. And RNA, the sugar on its backbone that alternates between the sugar phosphates, is ribose. So DNA has one less oxygen on that sugar than the RNA sugar. So the sugars are a little bit different. And that's partly why the uracil is used instead of the thymine. It's an easier fit to bond with the sugar on the RNA molecule. So after transcription happens, after we get this transcript, this RNA copy of the DNA, we have some things that take place after that, before we're done. It's called post-transcription modifications. You can think of it like a rough draft versus a final draft. Initially, we get this nice little messenger RNA molecule that gets spit out from our transcription process. Then it looks like this. It's sort of the rough draft version, the pre-mRNA. What it's made of things called exons and introns. Exons are the ones that actually get used. Introns are the pieces on the RNA molecules that get cut out by enzymes. Enzymes come along and they cut here and they take these pieces out. And it's called excised. Those pieces are excised, and you can kind of see here. Then the exons scoot together like this. Those introns get kicked out, and we have the mature mRNA. The messenger, M stands for messenger right there. Messenger RNA piece, that is the piece that will end up giving us our final product, our protein. One more post-transcription modification that is made. After this getting rid of the introns, there is a cap that is placed on the front end of our messenger RNA molecule and a little tail, a poly-A tail that is placed on the end. And that completes our RNA, piece of RNA that will go and perform the things that it needs to perform. Translation is our next process, and we'll talk about that in the next video. And that translation then will make a protein. So one more thing we need to talk about. We need to talk about some strange thing called the central dogma. Dogma is just like a rule that can't be violated, sort of a code of ethics, something that is a rule. So the central dogma in genetics happens to be one gene makes one protein. This is sort of the way we used to always think about genetics. We thought that DNA, it can replicate itself here through the process of replication. It makes, through transcription, makes RNA. Then RNA, through the process called translation, makes a protein. We'll talk about that in the next video. We haven't talked about that yet. So this is the central dogma, that DNA makes RNA, makes a protein, and that one gene of the DNA will translate into one protein. It isn't always how things work. Sometimes we have found in modern genetics, sometimes one gene does not determine one protein. Let's look at a few different violations of this rule. So the not so central dogma, one thing that can happen, remember when we talked about introns and exons, we can actually have alternative splicing where maybe this gene, if it's spliced in a certain way, will produce this protein. Or if it's spliced in a different way, it can produce this alternative protein, protein B. We can have that happen. One gene could actually produce several different proteins. Some functional parts of genes don't even make proteins. They make things like other pieces of RNA, other types of RNA. They don't make proteins at all. Sometimes there is even really strange things can happen where you get reverse transcription, where RNA goes back and makes DNA. The HIV virus is an example of where this is caused. So we see things that are not part of our central dogma. They sort of violate this rule. However, most of the time, the central dogma does hold true. Most of the time, one gene codes for one protein. Just that it's not always that clean and nice. It is a little bit messier than that in reality. So here are some advanced ideas. If you want to go for advanced proficiency, look at what is the structure and function of those different kinds of RNA. Look at that tRNA. What does that do? What's that all about? There's several different kinds of RNA. What is the role of spliceosomes? I always thought that was sort of a funny sounding word. Those are enzymes. What do they do? What enzymes are involved in transcription? You can go and look up the specific names of these enzymes and explain how they're involved in the process of transcription. And coming next, in our very next video, we have the process of translation. We are going to use this messenger RNA that we've created through the process of transcription to assemble proteins. And we'll see what that looks like in the next video.